There you go. All right, so just to give you a heads up as far as what is happening. So starting today, we'll cover male reproduction. We should be able to get through most, if not all of it. Next Tuesday, this reproduction 2, which is female reproduction. We will have an exam review, and I will be sending out a study guide for that sometime over the weekend. Next Thursday, so that's a week from today, will be our last exam. It's going to be worth twice as much, just because a lot, bless you, a lot in reproduction. So it'll be worth 100 points. I'm not sure as far as the breakdown. It'll probably be mostly multiple choice again, with maybe a few more essay. I might mix in a few other things, but I'll let you know. And that's also the date that the extra credit is due. So some of you have already done the extra credit. That was given way back in the beginning when I forwarded you that video on that guy talking about COVID. That was one. And then the second one was the more recent one on Gesundheit, cystic fibrosis, understanding the different transport mechanisms with that. And there, of course, a couple different transport mechanisms were introduced that we didn't cover in class. I hope to have everything pretty well done by June 1st to post the grades and everything. I am starting off teaching summer school at SDSU, actually starting next week. So they actually start the middle of next week. So it's going to be a little crazy, but
it must be very interesting.
Yes. Yes, it is. All right. So let me go ahead and start with today's lecture. Okay. So the male reproductive system. Now, the male reproductive system, actually reproductive systems in general, are really divided up into two parts, what we call the primary sex organs, or call them the gonads. That would be the testes and the ovaries. And then we have the accessory organs, which consists of glands that help, for example, provide nourishment for eggs and sperm, help in transport, and also external genitalia, which have a protective role. Now, obviously, we've spent a lot of time on the endocrine system. And, excuse me, I had mentioned to you that we would cover more of the endocrine, the reproductive aspects of the endocrine system we'll cover during reproduction. We really didn't focus much during the endocrine section, just in name only, but just to give you a bit more detail. The endocrine system with both males and females operates basically the same as far as many of the hormones involved. There are some differences. Some of the common features are the fact that the master orchestrator, the controller, is the hypothalamus, which produces GnRH. That arrow is a little bit close, but we see an increase in GnRH, which travels to the anterior pituitary, which triggers the release of both FSH and LH. Now, those two hormones, they go to different, first of all, the target, of course, is the testes in male reproduction. But where they go in the testes are two different places. FSH travels to what we know as the Sertoli cells. We'll talk about those today. These cells are very important in providing nourishment and protection to allow for spermatogenesis. And they also produce a hormone called inhibin. One of the things we've learned about in the endocrine system is the role of negative feedback. Well, we have two hormones, FSH and LH. Inhibin inhibits FSH secretion. So this would be kind of a long loop feedback from the Sertoli cells all the way back to the pituitary. What about the role of LH? LH travels to the Leydig cells in the testes, and these cells are involved in the production of testosterone. So you can see the increase in testosterone. What does testosterone do? Testosterone plays a role in a lot of different secondary male characteristics, hair growth, growth of reproductive organs, voice changes, things like that. But it also has more of a local effect in also promoting spermatogenesis with the Sertoli cells. So overall, FSH, what are the roles of FSH and LH? In males and females, well, first, let's just talk about males. In males, it promotes spermatogenesis, gonadal hormone secretion, that would be testosterone. So production of gametes, because sperm is a gamete, production of gonadal hormones such as testosterone. And thirdly, the effect of LH and FSH on gonadal structure that occurs during development and puberty. All right, so now we're going to take a journey through all the different components of the male reproductive tract. And as you can see here, several different organs are listed. I always like to start with the testes. So first we're going to talk about the testes, which is the site for spermatogenesis. Next, we're going to travel through this structure, which is known as the epididymis, and we'll talk a bit about that, its role in maturity and storage of sperm. Sperm mature a bit more in the epididymis, and they're stored here. And then transport of the sperm through the structure, which has two names. It's either known as the vas deferens or the ductus deferens. I think the more common use is the vas deferens. If you've ever heard of, most people have, of a vasectomy as a contraceptive method. There's a suture or a cutting that occurs here, and it's sutured that prevents the sperm from traveling out to possibly fertilize an egg. So this is the vas deferens. It carries the sperm into the pelvic cavity. And of course, as we know, the testes are located outside the pelvic cavity. And there's a very good reason for that we'll talk about. A lot of it has to do with temperature. So from the testes, the epididymis to the vas deferens carries this into the pelvic cavity. And if you can picture the sperm traveling through here, right around this point, this area, we see this structure known as the seminal vesicle. This is an accessory gland. This is probably the most active of accessory glands. We'll learn a bit as far as what it produces. One of the things it does make is fructose, which is an energy source for the sperm, as well as other substances. And the vas deferens secrete substances through this duct. And here we have this, again, the vas deferens coming down. Here's the seminal vesicle squirting its contents into the duct. What was known as the vas deferens is now known as the ejaculatory duct, as it merges with the prostate gland. So the prostate gland is kind of a walnut-sized organ. And you can see by its location, it's just underneath the urinary bladder. And one of the things, and we'll talk about this later, that can occur in males as they get older is the prostate gland enlarges and it will compress the urethra. Remember the internal urethral sphincter and the external urethral sphincter. Well, prostate can actually compress and cause problems. Now, as far as its main principal role, much like the seminal vesicle, it also produces substances for the sperm, nutrients. It actually produces a very alkaline fluid or very basic fluid. And that's very important because if the sperm make it into the vagina is very acidic. I know if whoever is, if who's ever sneezing a lot, if you can like, yeah, mute, maybe I can mute it. There we go. Okay. So that's the prostate. Produces alkaline substances to protect the sperm against the acidic vagina, as well as it also produces other nutritive material. The sperm continue on past the prostate into what's known now as the urethra, where they merge with this organ known as the bulbourethral gland. That's the last of the 3 accessory glands. And that produces what sometimes is called a pre-ejaculate. It produces a lubricating substance, which helps to facilitate the movement of sperm through the urethra. And of course, this part of the urethra moves through this other structure, which is the penis, which, as we'll learn, is an erectile organ, and we'll see why it is. All right, so I spoke about the testes, and one thing that I brought up is I mentioned that temperature is really key, and the fact that they're located outside the pelvic cavity. Well, of course, the testes are located outside the pelvic cavity, but they're contained within a sac known as the scrotum, which is this right here, which surrounds the testes. Within the scrotum, and actually connected to the testes, a couple different structures are really important in terms of what we call thermoregulation. Now, why do we need to thermoregulate to control the temperature of the testes? It's because that spermatogenesis is optimal, the process is optimal roughly at 2 to 3 degrees cooler than the rest of the body. And in order to achieve that, number one, we have the testes located outside the body cavity, outside the pelvic cavity. But we also have these blood vessels that we'll talk about called the pampiniform plexus, kind of a tongue twister, that's a vein, and the testicular artery. So we're going to talk about those 2 vessels. And third, what's known as the cremaster muscle. Those 3 structures, the 2 vessels and the cremaster muscle, are going to be responsible for maintaining the cool temperature of the testes within the scrotum. So how does this work? Okay, so here you can see a close-up view of the veins. And of course, as typical with veins, in this case, the veins will drain the testes. There's part of the systemic circuit. And the red, the testicular artery, supplies the testes with warm, oxygen-rich blood. And the veins drain it, and they're typically cooler, okay? Well, as you'll see on the next slide, the arrangement of the artery and vein are such that as the arterial blood enters into the testes, it's warmer, it's coming from the body core. It transfers heat to the veins. And now the veins are leaving warmer and the arteries are arriving cooler because of that heat transfer. That helps to keep the testes cooler. The other structure is this cremaster muscle, which can change the position of the testes to account for changes in ambient temperature. So if it's colder, they raise the testes. If it's warmer, they lower the testes. So those are the 3 structures involved in thermoregulation within the scrotum. I did want to mention another structure that encompasses some of these. This, what's called the spermatic cord, this is a structure that's surrounded by connective tissue, but it contains blood vessels and nerves and the vas deferens. It's called a cord because it looks like a cord that's leaving the testes. It contains the testicular artery. It contains the pampiniform plexus. It contains the vas deferens, which is kind of hard to see, but there it is right there. And it also contains autonomic neurons. So the testes are regulated by both the parasympathetic and the sympathetic systems. All right. So as I mentioned, the arterial supply of the testes is the testicular artery, which is a branch off the aorta. The veins are the pampiniform plexus, which ultimately enter into the vena cava. And as I mentioned, the testes are innervated by the ANS. All right, so I wanted to give you a little visual of how thermoregulation works. This, of course, is the spermatic cord. And you can see all the different structures that are part of that. We have the arteries, the veins, the ductus deferens. You can't see the nerves here, but they're there. This is what we call the countercurrent heat exchanger. This in red is the testicular artery. This in blue is the testicular or the pampiniform plexus. And you can see how, follow the arrows, heat is transferred from the arteries to the veins. So imagine blood arriving, going towards the testes, that's about 37 degrees, which is our normal physiological temperature. By the time it reaches the testes, it's about 35. All right, so one last view, kind of an overview of the reproductive system before we start kind of jumping into looking at the anatomy and physiology of each region. So here, this is a frontal view, obviously. Testes, epididymis, ductus deferens, seminal vesicle, prostate. Here is the urethra, bulbourethral glands, and this tissue on either side of the urethra, that's the penis. All right, so let's start off with the testes. The testes of the male, much like the ovary of the female, are the factory. This is where a lot of the active production of gametes occurs. So testes, of course, are the site for spermatogenesis. If we look microscopically, sorry about that. If we look microscopically, or at least at a lower magnification, higher magnification, we could see these squiggly tubules in the testes, which is kind of cut away. These are what are called the seminiferous tubules. Within the testes, which is this right here, seminiferous tubules are the site for spermatogenesis. This is a light photomicrograph showing these circular tubules. This is a higher magnification. So this is where spermatogenesis takes place. And I'm going to show you a bit more detail microscopically what the seminiferous tubules look like, close up, and some of the other contributing factors. All right, so this is a testes from the cadaver. You can see the testes, the epididymis, as well as the ductus deferens. All right, so microscopic anatomy of the testes. So we have these seminiferous tubules. What's inside these tubules? Let's kind of zoom in. Within these tubules, we have what are called spermatogenic cells. Spermatogenic cells are cells that are destined to become mature sperm. And we have all stages of development, from stem cells to fully mature sperm cells that look like this, the typical structure that you're familiar with seeing. So we have this abundance of developing spermatogenic cells, and we'll take a look at them up close. In between, interspersed among the spermatogenic cells, we have what are called Sertoli cells. Remember, I mentioned that early on. The Sertoli cells are the targets for FSH. And it's these Sertoli cells that are there to make sure the testes, the spermatogenic cells are have an adequate nutrition, they're protected, and it helps to ensure that spermatogenesis proceeds. Now, yes, we've got a lot of these seminiferous tubules, but in between seminiferous tubules, so this is a tubule, this is part of a tubule, this little area, which kind of looks like a triangle, these are known as Leydig cells. They're found in what's known as the interstitial tissue. Interstitial tissue is between the seminiferous tubules. These Leydig cells are the source for testosterone. This is where testosterone is produced. And as we'll see a little bit later, that testosterone, being a steroid, a lipid-soluble substance, travels across into the seminiferous tubules where it helps to also ensure that spermatogenesis will continue. The production of testosterone is under the influence of LH. There is another group of cells that quite often aren't mentioned in the literature, but these are smooth muscle. We call them smooth muscle-like cells because they are not exactly true smooth muscle, but they function like it. They're called myoid cells. And they contract just like muscles do, which helps in moving the sperm outside of the seminiferous tubules once the sperm are produced. All right, so this is kind of the big picture here. This is a nice view of a seminiferous tubule. Here we've got all these spermatogenic cells. You can see you can't see the details, but you can see a lot of different levels of nuclei, right? We also have Sertoli cells, which are really a supportive cell, providing nourishment and protection to the developing sperm. In between seminiferous tubules, we have these Leydig cells, which are these produce testosterone. And then we have this smooth muscle-like tissue which surrounds the seminiferous tubules. These are the myoid cells. All right, so let's zoom in on spermatogenesis. What do we know about it? Well, we know, of course, that it's the process of immature sperm becoming mature sperm, which are capable of fertilizing. Roughly 400 million sperm are produced by human male per day. This process begins at puberty, which is fascinating in the sense that with females, the production, the development of eggs in females starts occurring when that female is still a fetus. So when a female is a fetus inside her mom, those ovaries are starting to produce some eggs. Now, obviously, those eggs don't reach maturity yet. Ovulation doesn't occur until puberty, but at least the eggs are starting to develop. In males, the development of sperm doesn't occur until puberty. And it takes about 75 days to go from the least mature sperm cell, which, by the way, is called a spermatogonia, towards the most developed one, which is the spermatozoa. So if we take a look at the process, let me go back to the previous slide for a second. One of the things that we know is that spermatogenesis begins on the outer part of the seminiferous tubule. This is called the basal side. This is where we have spermatogonia. And the development of the sperm progressed into the lumen. Okay, so let's take a look at this picture. This is the basal side, this is the lumen. We start off with spermatogonia. These are the immature cells, and they undergo division. We'll talk about the type of cell division to become primary spermatocytes. I like this picture because it's color-coded, which is nice. So spermatogonia here, primary spermatocytes. Those primary spermatocytes undergo division to become secondary spermatocytes. Those secondary spermatocytes undergo division again to become what are called spermatids. And that's where the cell division stops. The spermatids transform or differentiate into a mature sperm cell, which are these, just by morphological changes. No more cell division, but incredible transformation from one cell to the other. All right, so let's talk a bit about, so we've spoken about the spermatogenic cells, the different types. Now let's talk a little bit more about these Sertoli cells, because they have a lot of very important functions that it's worth talking about. First of all, they surround, or they're in between spermatogenic cells. This is one right here that's kind of squeezed in the middle. Here's another one. They're pretty long. They extend from the basement membrane or the basal area all the way to the lumen. Blood vessels are out here. Those Sertoli cells help to move nutrients into and nourish the spermatogenic cells. So they're kind of a conduit for transporting nutrients. And they help in the movement of sperm cells into the lumen as well. Another important function, so they have an important role in spermatogenesis, in providing nourishment, but they also provide a more important role as well. Remember the picture I showed you of what an immature sperm cell looks like and what a mature sperm cell looks like? That's a big difference, right? And some of the changes involve loss of cytoplasm. Those Sertoli cells are responsible for phagocytizing the cytoplasm of the sperm, so that all that's left is the nuclei, some mitochondria, and a tail which develops. So, and we're going to take a look at that process as well. A little bit more about the Sertoli cells, and I want to spend a lot of time because they really are important. Along the lines of them being protective and providing nourishment, they also help to maintain what's known as the blood-testis barrier. We haven't spoken about that, right? We've spoken about the blood-brain barrier. Blood-testis barrier is similar in the sense that it prevents certain substances from crossing over into the tissue, substances that could be damaging, toxic. In this particular case, it also prevents the immune system from gaining access to the spermatogenic cells. And the reason for that is interestingly, the immune system recognizes the sperm cells as foreign. Why is that? Because they have 23 chromosomes, because they're shaped differently, different antigens, I don't know. But if immune cells like lymphocytes were to get access to the seminiferous tubules, they would destroy the sperm. So someone would become sterile. So they have to maintain this barrier. They also secrete a lot of things. They secrete nutrients, obviously. They also secrete a substance known as androgen binding protein, which helps to concentrate the testosterone that's produced by the Leydig cells and concentrates it inside the seminiferous tubule to keep it there, so it allows the testosterone to exert its effects. Now, this is not a typo, spermiogenesis. I'll talk about that shortly, what that is. Those same Leydig Sertoli cells also produce a couple different substances. They produce inhibin, which you know about already, which is a feedback loop that inhibits FSH. But they also, interestingly, produce activin, which stimulates FSH. So inhibits, activates FSH. And as I mentioned before, the phagocytosis of the cytoplasm of these spermatogenic cells as they're approaching maturity. Oops. All right. So this is meant to show the blood-brain barrier, blood-testis barrier. So this is the basal part of the testes, right? This would be the lumen. This is where the blood vessels are. This is where testosterone is. You can see just the arrangement of these Sertoli cells. They actually form not only a barrier in terms of well, not only do they nourish, but they actually form a physical barrier that prevents anything that crosses through the basement membrane from accessing the cells. Now, it looks like they might be able to access them here. This is probably not the best drawing, But suffice it to say that Sertoli cells basically wrap around the developing sperm cells to prevent toxic substances, to prevent the immune system from getting access to the seminiferous, to the sperm. All right, so as we know, the other two cells, just to reiterate, we have the myoid cells, which contract, and we have the Leydig cells, which are known as, some books, and I put this in here just to avoid, hopefully it won't make it more confusing, but interstitial cells is just another name for Leydig cells. Same thing, they produce testosterone under the influence of LH. All right, so let's take a look at spermatogenesis. I found that last time that this video doesn't work because of some flash issues, but we're going to cover it pretty thoroughly anyway, the process. All right, so what do we know about spermatogenesis? Spermatogenesis, as I mentioned already, is the process from where the least mature sperm cell, which is often known as the spermatogonia, eventually develops, changes into the mature sperm cell, which is called the spermatogonia. But on the way, it changes, right? There's several different steps. The first step is spermatogenesis, spermatogonia dividing to form primary spermatocytes. And this is through mitosis. Doesn't show it up here, but this is through mitosis. Primary spermatocytes become secondary spermatocytes through meiosis I. And secondary spermatocytes become spermatids through meiosis II. The final step is no more division, but through actions of the Sertoli cells and other mechanisms, the spermatids change into spermatozoa. All right, so this is a much better picture. You can really see the extent of it. And I wanted to add a couple elements to this. Of course, we know, as I mentioned, that spermatogonia divide to form primary spermatocytes, and ultimately meiosis I into secondary spermatocytes, meiosis II into spermatids, and then finally, the last step, spermatids becoming spermatozoa, or mature sperm cells, is a process known as spermiogenesis. The whole thing is called spermatogenesis, but the last step from spermatids to spermatozoa is called spermiogenesis. Now, I wanted to mention another key point. We have all these stem cells in the seminiferous tubules, right? These spermatogonia. If all of them divide to form spermatocytes, eventually we're going to deplete the spermatogonium. Well, fortunately, what happens is when the spermatogonium divide, they actually will form another spermatogonium and a primary spermatocyte. So basically, they have what we call type A daughter cells and type B. Type A daughter cells are making more and more spermatogonia. Type B daughter cells becomes primary spermatocytes. So that's a way to ensure that we're sustaining the amount of stem cells that are necessary for this process to continue. All right. Spermiogenesis. Spermiogenesis is the maturation of spermatids into spermatogonia. And you can see it's pretty dramatic. Going from a cell that looks like this, like a normal cell, into one that looks like this. Well, we know that there's 4 steps involved, what we call the Golgi phase, the cap phase, the acrosome phase, and the maturation phase. I'm not going to ask you to know details about those phases, but just know the names and the order that they exist. What are some of the real obvious changes that we're seeing? Well, first of all, one clear thing is going from this cell to this cell, we lose the cytoplasm. So the Sertoli cells phagocytize the cytoplasm. Now, when they phagocytize it, everything is removed except for a couple things. The head, which is a nucleus, So the nucleus is left, right? The mitochondria, these structures right here that are eventually aggregating right in the mid-piece, that's mitochondria. Mitochondria, of course, are the energy currency of the cell. They're essentially the motor that allows for the spermatozoa to move. That's the mid-piece. Okay, so we see the development of the mid-piece, which is the accumulation of mitochondria. We see the development of the tail, which is the flagellum. We have the nucleus, which I forgot to mention, that's known as the head. But we also have this other structure, which looks like a cap. This is the acrosome. So we have in the mature sperm, we have the acrosome, the head or the nucleus, the midpiece or the mitochondria, and the tail or the flagellum. All right, let's take a look at these a little bit more up close. Spermatid, we can see what happens. We see the clustering of the mitochondria. We're starting to see the eventual removal of the cytoplasm. We don't see the Sertoli cells, but they're doing their job. And we start to see the mitochondria lining up. And we see, of course, the development of the tail. And we have the head and we have the acrosome. All right. Okay. Oops. Okay, so to kind of summarize a bit, spermatogenesis occurs at puberty. We haven't talked about chromosome number. I know you've learned that in other classes, but I did at least want to mention that during this process, we take a diploid cell or a cell that has 46 chromosomes, the spermatogonia, and it eventually becomes a cell that has 23, right? And that mature cell that has 23, if it fertilizes the egg also has 23, so 23 and 23 is 46. We have a fertilized egg with the full complement of chromosomes. We have mitotic divisions, which can form type A spermatogonia, which will make more spermatogonia. We have type B spermatogonia, which will ultimately become primary spermatocytes. After that, we have 2 rounds of meiotic division of the primary spermatocyte, which will ultimately become a spermatid. Remember, primary spermatocyte becomes a secondary spermatocyte, becomes a spermatid during meiosis. The last process, spermiogenesis, is the morphological differentiation of spermatids into a mature spermatozoa. Notice I didn't say divisions because there are no division. It's just a change. And that change is also evident right here. This is part of the sperm. You can see this would be the tail portion, which is this. This is the mid-piece, the mitochondria. These are mitochondria. This is the head, the blue, the nucleus. And then we have the acrosome that sits on top of it. So what are the significance of these regions? Well, of course, the head is the nucleus, condensed nucleus. It contains the DNA, which is important for delivering to the egg to combine with the DNA from the egg. The acrosome is a very important structure during fertilization. It contains a packet of digestive enzymes, which helps to break through the cell membrane of the egg, makes it more porous, which allows the egg to be fertilized. If there are some causes of infertility, obviously a variety of causes in both males and females, but one cause is maybe a defect in the acrosome, which a sperm might reach the egg, but if the acrosome isn't working properly, and we'll talk about that next time, the egg won't be fertilized. All right, so we've made our sperm, right? We've completed spermatogenesis. Next destination, sperm have to leave. They're getting booted. So from the seminiferous tubules, Remember I mentioned that the sperm eventually make it to this structure, which is the epididymis, right? And then eventually to the vas deferens. Well, en route from the seminiferous tubules to the epididymis, there's two structures the sperm has to pass through. One is known as the rete testes, and then eventually into what we call the efferent ductules, and then the epididymis. So I'm just adding a couple more elements of transport from the seminiferous tubules to the rete testes to the efferent ductules, and those myoid cells are helping to contract, which is helping to push the sperm into the efferent ductules and eventually into the epididymis and the ductus deferens, where their sperm are now on their way to the pelvic cavity. All right. So, but before we go to the pelvic cavity, the sperm actually pause in the epididymis. The epididymis has a couple important functions. First of all, the sperm that have left the seminiferous tubules, even though they might, under the microscope, basically they might look like a normal sperm, they're not ready to fertilize an egg. They need to undergo some biochemical changes, improve on their motility. And the motility is the movement of a tail. And that is really a very important part of fertilization. I mean, another cause of infertility is the tail isn't working properly. So this is kind of a pit stop, so to speak, where the sperm, again, get their metabolism enhanced, biochemical changes, they get their motility, and they're stored here prior to ejaculation, which is then going to move the sperm into the ductus deferens. All right. So this is the epididymis, as you can see right here. We've covered a lot of, well, you guys know some histology. The histology of the epididymis is somewhat unique in the sense that it's pseudostratified columnar, not pseudostratified ciliated columnar, which is what we see in the trachea, right, in the bronchi, but pseudostratified non-ciliated columnar. Now, it does have what are called stereocilia, which is just a large concentration of microvilli that they don't move. Remember, cilia, they sweep. These don't move. They just stand, not stand, but they just remain motionless. All right, so from the epididymis, the sperm then are transported into the vas or the ductus deferens, which is this more of a straight structure as opposed to coiled. What do we know about this structure? This is a highly muscular organ, or well muscularized. Contains 3 layers of smooth muscle. It contains a longitudinal layer, contains an inner longitudinal, a middle circular, and an outer longitudinal. So a lot of muscle, and of course muscle contracts. This is important in the sense that this allows the movement of sperm during ejaculation through the vas deferens. The histology is also pseudostratified columnar epithelium. And it has this outer adventitia, which is kind of like a serosa, but it's different. So just know it's a very muscular organ, as opposed to the epididymis, which has the epithelium, but this is more for storage and development of the sperm, as opposed to movement through contractions. This is kind of doing a little comparison contrast between the epididymis. You can see these very tall looking cells. What you're looking at here, these are all sperm in the lumen, and these are the stereocilia on the outside. This is the ductus deferens, and we have, like I mentioned, an inner longitudinal, middle circular, and an outer longitudinal layer of muscle. Sorry about that. All right, vas deferens. Now, I mentioned before, one term that's often used surgically with vas deferens is vasectomy. This is the surgical ligation or cutting and ligation of the vas deferens, which doesn't affect sperm production, but it does affect sperm transport. So people often ask, well, what happens to the sperm? What happens? They're eventually reabsorbed. And since they're not being transported, they end up being reabsorbed and broken down. This process does not affect testosterone production or sperm production, but it certainly does affect the ability to fertilize an egg. This is, it says here, this is interesting. It says that it should be considered a permanent birth control method. Having said that, however, there are certain types of vasectomies that are done that can be reversed. My uncle, in fact, had this done. He had a couple kids. They decided that him and his wife decided that they didn't want any more. He had a vasectomy, and a year later, bam. So obviously the vasectomy didn't work. But anyway, he eventually had it done again. All right. So just retracing our steps, testes, through the epididymis, through the ductus deferens. Now we're in the pelvic cavity. And if you want to follow, this is the ductus deferens, both sides. Remember, one from each testes. As they enter into the pelvic cavity behind the bladder, right? The next structure along the way are these two. This is just a slice through so you can see the inside, but these two are seminal vesicles. So these are the first of the accessory glands. The second accessory gland is this one, which is the prostate. And then finally, we have the bulbourethral gland. So we're going to take a look at each of these three. All right, what do we know about the seminal vesicles? Well, if you look at the production of fluid from each of these glands, because that's what glands do, right? And because they're secreting into ducts, these are exocrine glands, accessory glands, exocrine accessory glands. The seminal vesicle supplies about 70% of the fluid. Now, once the sperm encounters this fluid, now it's known, the whole material is now known as semen. So semen refers to sperm plus the contributions of the accessory glands. What do we find in the seminal vesicles? We find fructose. We find a variety of substances, including prostaglandins, that help to increase sperm motility. Because if you think about it, the sperm have traveled all the way from the testes, they're working their way up. They need a little energizing. They need a little energy drink. So they get their energy drink here from the seminal vesicle. And also, too, there's a variety of substances that suppress the immune response against semen. That's kind of not clear as far as how that's done. But the three that we do know about for sure are these first three. Second of all is the prostate, which we often hear more often about just because of its involvement in certain types of diseases. This produces about 20% of the volume of semen. Again, motility is really crucial here. So some of the substances from the prostate help to enhance motility. And also substances that are involved in the clotting and liquefaction of semen, which does occur in the female reproductive tract. The pH of the prostate fluid is very alkaline, which helps to protect the sperm against the acidic conditions inside the vagina. All right, pathologies. So benign prostatic hyperplasia, it's also known as benign prostatic hypertrophy, is really the more common. This is an enlargement of the prostate due to the increase in the number of cells. So hyperplasia would be an increase in the number of cells. Hypertrophy would just be an increase in the size without an increase in cell number. So here we're looking at an increase in the number of cells. This is a non-malignant condition. That's why it's called benign. This can affect males. Well, it does affect males that are typically older, and affects roughly 75% of men that are over 80, which is pretty substantial. Now, what are the complications? So it's not cancerous, but due to its location, would be a normal prostate. This is the urethra, right? As the prostate enlarges, it pushes on the urethra, which affects the ability to urinate. Now, in some cases, it becomes difficult to urinate as far as reduces the stream, but it does increase the urgency. So once someone does have to go to the bathroom, normally, there's a certain amount of time. It's like, yeah, you sort of feel like you got to go, but you're not near a rest stop, you're not near a bathroom, you're fine. With people who have a severe hyperplasia or even hypertrophy, once they get that sensation to go, they got to be near a bathroom. So there's a sense of urgency. Then there's prostate cancer, which is the most common cause of death from cancer in men over 75. The incidence of prostate cancer is roughly the same that it is with women with breast cancer. I believe it's like 1 in 8 or 1 in 9. And of course, it's malignant. So meaning that the cancer will spread into other organs of the body. Last exocrine gland, accessory gland, is the bulbourethral gland. It's very small. It does produce the smallest amount of fluid. It produces kind of a mucus lubricating fluid that helps in facilitating the movement of sperm outside the body. And it does also add a bit more alkaline. The mucus is on the alkaline side, so if the sperm has any acidity to it, which it can a little bit, if it wasn't taken care of by the prostate, the bulbourethra does. Now, you see CalPERS, I haven't mentioned that before. As we know from many terms that we've spoken about, that there's more than one way of saying the same thing. Bulbourethral is another name for CalPERS. That was named after the person who first discovered the gland. All right. So we're almost at the end of the story. And if you follow the path of sperm from the testes to the epididymis, to the ductus deferens, into the urethra where it gets contributions from the seminal vesicle, the prostate, the bulbourethral glands. Now the sperm is still in the urethra, but it's now passing into the penis. And there's a couple of different parts of the penis, the shaft, what we call the glans portion, and the prepuce, which is found in uncircumcised males. It's the foreskin. All right, so what about the detailed anatomy of the penis? Remember I mentioned it's an erectile tissue. And when you talk about erectile tissue, it's a tissue that gets engorged with blood. Comparable organ in females would be the clitoris. In this particular case, the erectile tissue are these two, which are called the corpus cavernosum. This one down here is a similar type of tissue, but it does not fill up with blood. It's surrounding the urethra, so we don't want any changes in it. This is called the corpus spongiosum. To put a little humor in this, when you look at this picture, it kind of does look like a face, 2 eyes and a mouth. All right, so if we take a look at the male reproductive response, there are 3 parts to it. They all start with E, erection, followed by emission, followed by ejaculation. Erection involves the dilation of arterioles, so this would be the arteriole, right? Which leads to leakage of blood into these spaces. These are all spaces. This is under parasympathetic control. Remember I mentioned that there's autonomic neurons innervating the male reproductive organ? All right. This leads to erection. Following erection, there's what we call emission, which involves the contraction of the epididymis, the vas deferens, secretion of accessory glands, and this is under sympathetic control. So here we're starting to look at the movement of the sperm, right? Here we've got the erection. The sperm begins to move from the epididymis. We start to get contributions from the glands. This is this is sympathetic control, not parasympathetic. And then what continues after emission is ejaculation, where the sperm has now moved past the vas deferens. They're in the urethra contracts. This is a really important process right here. There's also a simultaneous closure of the internal urethral sphincter. So if you remember in our discussions of the urinary system during micturition, remember we have the internal sphincter relaxes and the external sphincter relaxes, correct? And this allows urine to go out. Well, during ejaculation, we want the internal sphincter closed because that's going to prevent any semen potentially from going back. So if I've got a picture, and this doesn't, let's see. It's a good picture. No, let's see if I can find a good picture. Sorry about this. Yeah, this is pretty good right here. Actually, no, it's even better. There we go. All right. So here's the bladder, right? This is this duct. We would have sphincters right here. Well, if you imagine sperm that's going out this way, right? We normally have this sphincter right here. If this sphincter was sphincter was closed. Wait, hold on a second. What am I talking about? I'm sorry. I'm screwed up. The internal sphincter is up here. If this sphincter was open, some of the semen could back up. But by closing of the sphincter, this prevents sperm from going up, potentially into the bladder. Yeah, so closure of the internal urethral sphincter. All right. All right, so let's take a little closer view at kind of the process of erection and how it occurs kind of physiologically. I've got a couple of different diagrams to show you. First, I'll talk about it right here. First of all, there's nerve involvement. Of course, as we know, it's the parasympathetic nervous system. And of course, what triggers this? Obviously, sexual thoughts, other activities, whatever. Stimulates, the nerve stimulates the arteries, which causes them to vasodilate, which is the relaxation of the tunica media of those arterioles. This results in increased blood flow into the cavernosum. So this is in an unstimulated penis. This is in a stimulated penis. You can see not only is blood in there, but it's enlarged, the organ. Now that's one thing, and that's important for erection. But one thing that allows the erection to be sustained is at the same time the arteries are dilating, allowing blood to go in. The veins, which are located on the periphery, are being compressed. So you have an increased inflow with a reduced outflow. And this allows for the sustaining of the erection. And finally, a little bit more detail about the urethra. I did want to mention that, of course, the urethra extends from way up here, just outside the bladder, all the way to the end. It's all the same structure, but it has regional differences. The first part is called the prostatic urethra because it does go right through the heart of the prostate. The part that goes through the penis is sometimes called the spongy urethra. And in between the small little segment is called the intermediate part of the urethra. All right. All right. So that takes us to the end of the first round of the reproductive system. Thanks for showing up. Not sure if anyone has any questions. I could leave it out there in case anybody does. If there are no questions, we'll go ahead and end it here. I will be posting the video again, as usual, online, or I'll post it on YouTube, and I'll send you the link. Let's see. I will be updating the second reproductive lecture. So if you've already started looking at it, the second lecture, that's fine, but just note that it's not going to be the same in a couple of days. I'm going to modify it. And I will also be sending you guys out a study guide. And that's it. So any questions? All right. Well, I will see you guys next Tuesday. Again, if you have any questions, feel free to contact me.